Well, thanks everyone for tuning into this. This is a presentation I gave during BTME in Harrogate in January 2019, and it's disease suppression without fungicides. Can it be done? Now obviously disease control was always a problem for greenkeepers and course managers, but with the reduction of available fungicides, it's become even more important now in the last 2 years. So I'll just give you a bit of background to myself first of all. I'm basically a greenkeeper. I'm the course manager of the Royal Curragh Golf Course in Ireland. This is Ireland's oldest golf course. It's there since 1852. Not there quite that long myself, but at times it does seem like that. Anyway, as an inland course with poor bent greens, disease is a problem I've had to deal with for many years. Now, having been a greenkeeper for many years, I decided later in life to go back into education, and I did my degree in turfgrass science at Myerscough College in the UK. Something I would recommend to anyone starting out in the business. Gives you a very good understanding of what we're dealing with. And then when I finished that, I actually went south to Bristol, where I did a PhD in plant pathology part-time. So I was still managing the Curragh Golf Course while working on my PhD. And the basis of the PhD was the suppression of Microdochium nivale by phosphite in cool season turf grasses. So a lot of the work today now would be involved from that. So like I said, today's talk is disease suppression without fungicides. Can it be done? So basically we're going to look at managing turf with limited dependency on pesticides. So actually I'm going to use a number of field trials I've done over the years as a basis for this talk, and along the way we'll be looking at ways as turf managers to reduce disease pressures, looking at various cultural properties, looking at nutrients, and looking at defence enhancements in our turf grasses. And we'll be using this disease pyramid to highlight the different processes we can influence. At the end then we'll look at the results of the field trials we did, but then we'll be looking at the methodologies and the means by which this suppression of disease came about, and I'll be explaining some of the processes we use to determine this. So the two most common pathogens we have to deal with in the UK and in Ireland certainly are anthracnose and Microdochium patch. Like between the two of them we'd have pressure from these right through the year. And how to suppress these without using fungicides obviously is the focus of this talk. Anthracnose, very much a stress related disease and can indicate a problem with the turf. Obviously if we can reduce stress on our grasses, well, that's going to go a long way to reduce the incidence of anthracnose. Microdochium, or Fusarium patch as it was always known as, again this infects all really cool season turfgrass species. It likes humidity, moist surfaces, high amounts of nitrogen, and as you can see from the photos, I'm sure everyone is familiar with it, but it can be very devastating and cause a lot of damage, especially through periods of low growth when recovery is very slow. So this is the first trial we're going to look at. This is one I was doing for my PhD, and this started in 2010 and ran through to 2014. So I would have started in September each year and ran through to March. And basically we had 3 turfgrass species. I think you can see them with the marker here, hopefully. We had velvet bentgrass, Agrostis canina canina. We had creeping bent, Agrostis solanifera, and we had a Poa annua greens type sward. So we had 3 turfgrass species, and they were laid out in randomized plots as you can see here. And the treatments we applied were, we had a phosphite application. We applied phosphite with a biostimulant. We applied a fungicide, and a fungicide and phosphate combination, and of course untreated controls. So these are the mean values of disease incidence on the trial plots over 4 years. And as you can see over here on the right, these are the control plots which received no treatments of the three species. And what you note straight away is that the use of phosphate here on the right reduced disease incidence by about 50% in all species compared to the controls. Also, the fungicide program here, which actually included chlorothalonil and iprodion, which are both gone now of course, was quite effective at reducing disease. But importantly, the combination of fungicide and phosphate had a very good effect on disease levels. You'd often see this combination of different compounds has this synergistic effect due to different modes of action. So over the 4 years we found consistently that phosphate actually reduced the incidence of microdochium. Now we're also currently running another trial which started in September 2018, again looking at microdochium nivale infection, and this is going to run until 2019 March. We're using, apart from phosphate, we're using a number of treatment combinations which were especially targeted to try to reduce the levels of disease. We're comparing these to control and fungicide treated plots. Here's some preliminary results from that, and this is from between October and January, October 2018, January 2019. As you can see on the left here, sorry, on the right, as you look at it, the control plots had about 50% incidence on them. The use of this polyphosphate, phosphate 30 product had a very good reduction effect on them, just over 20%. And it's this combination here of various products which were targeted to suit the particular environment and soil conditions at the Royal Corus sites, which also had a very good effect on reducing disease. We're actually tweaking this at the moment to add some other compounds to it to see can we get further reductions. The Instrata fungicide, as you would expect, was very good at reducing, but so far it's looking good reductions on the phosphate and this combination of compounds. This is just some photos from those plots from earlier, start of January actually, or December 2018, and you can see the clear differences between different treatment applications there. This third trial that we're looking at was actually from 2018 during the summer, and we're looking at the effects of treatments on anthracnose levels. Again, these trials ran from July 2018 to October, and we applied the treatments every 14 days and we assessed disease incidence weekly. The treatments were actually this untreated control here. We had a polyphosphate treatment here, and then this combination or this cocktail of nutrients and products which were specially formulated to reduce anthracnose. And then we compared all this of course to standard fungicide Banner Max. Again, we had some pretty good results using those treatments. Here actually is the better graph. This shows the mean levels of disease over that trial period last summer. As you can see, the control plots quite high, up to 20% incidence overall. The phosphate product here, very good reduction on anthracnose levels, but to me surprisingly the biggest result was this combination of nutrients and phosphate here, which reduced levels of anthracnose to the same level as the Banner Max fungicide. And we'll be looking at these three trials and results later on to see how we got these results. That's just the peak incidence of anthracnose last summer, the 28th of August, and you can see it was up over 35%. Again, very good reductions from these three treatments here. So before we get into what the results of ours and how we got our results, excuse me, we're going to look at ways in which we as turf managers can actually reduce disease by using this disease pyramid. Now, most people would be familiar with the disease triangle, where the level of disease here is influenced by the environment, the pathogen and the host plant, and the interactions between these three. I actually prefer the pyramid, where we have the extra element of time. Now, when I say time, I'm actually talking about the time it takes the pathogen to actually infect the plant, but crucially it's the time it takes the plant to recognize it's being infected, and very importantly, the time it takes to respond with very complex defense compounds. And we'll be looking at this and how we can influence this area later on. But in the meantime, like turf managers like myself need to look at this disease pyramid and see what areas of this we can actually influence to reduce disease pressures. So, in the environmental factor here, we're looking at things like dew removal, increased air movement and light quality, basically areas that will reduce stress on the turf, but also reduce the ability of these fungal pathogens to develop and grow. Regarding cultural practices, we're going to look at there's various practices we carry out on a regular basis which can have a great effect on disease levels. Rolling, sand top dressing, mowing and irrigation practices we're going to see have a very big effect on disease pressure. Regarding the excuse me, plant factors, obviously if we can use less susceptible species and cultivars it would be a big help, but balanced nutrient inputs will also be very effective, because like ourselves, if we're suffering from malnutrition or if we're overweight, we're more susceptible to disease. And it would be the same with plants. You have to have this balanced inputs to ensure that they're not too stressed and not too soft or susceptible to disease. But also with regard to the plant factors here, the use of compounds, apart from supplying nutrients to the plants, can also have a direct effect on disease levels, and we will see that again. And this part here of this time factor of the disease pyramid, we're going to look at ways in which these defenses can be either primed or enhanced during infection, and there's a number of compounds here which will have that effect on the plants. Now, this is the first bit of solid scientific data I want to share, and this is looking at the effects of dew removal on microdochium. This is from ICL. It's research carried out at the STRI in the UK, and basically they looked at this compound, this product Dew Smart, which was applied to the replicated plots, and they found that this blue line is the level of disease on the untreated plots. And they found that by removing dew on a regular basis, it was a very significant reduction on disease levels. So like we've been removing dew for generations, we've been told to remove it, it's good for the plant and will reduce disease. And here is the scientific data to support that fact. So whether you're removing dew physically by brushing or whatever, or by chemical applications such as this, dew removal will have a big effect on disease levels. The other cultural practices now we're going to look at is something that's carried out very regularly by all greenkeepers really. Rolling, top dressing, irrigation practices and mowing heights. These four treatments or methods are going to have an effect on your disease levels also. First one we're looking at is actually the effect of rolling on anthracnose, and this is from Rutgers University, and it looks at the effect of rolling on disease levels in Poa annua putting greens. As you can see here, this top line is the level of anthracnose on the unrolled plots, and you can see by using either a vibratory or a sidewinder roller, they have a good effect on reducing anthracnose levels. This is caused by the roller inducing a physiological response, a stress response, and it's almost like priming these defenses prior to infection. Another thing which we carry out very regularly also is sand top dressing, And this is used to firm up our surfaces and to ensure that they're smooth, but it has a direct effect also on anthracnose disease. These plots here on the left, as you can see, are quite infected. They receive no sand, whereas the sand treated ones look much better and higher reduction of disease on these. And what's actually happening here again is a reduction of stress to the plants. As you can see in this graphic here, the sand top dressing is actually helping to protect the crowns and the sheath of the treated plants. Again, they're protecting it. The sand is protecting these areas and reducing the stress. And as you know, as I said, anthracnose is very much a stress related disease. Irrigation practices also have a big effect on the levels of disease. Again, it's talking about managing stress in your turfgrass surfaces, and you can see the difference between this 100% ET replacement and 40%. So obviously, if you can get in between the middle area here, you're going to maintain your surfaces healthily enough and not too soft, but also it will be less susceptible to the very dry, stressed out areas also. So a medium path with irrigation, ensuring that there's good coverage and uniformity of coverage, will help to also reduce anthracnose levels. Again, this is fairly straightforward as well. This is looking at the effects of mowing height on anthracnose. So you can see this is the level of disease here at 2.8. Sorry, this one here, the top line is the level of disease at 2.8 mil. By increasing the height of cut to 3.6, there's a good reduction again on anthracnose levels. Obviously, you're reducing stress again on the plant. And that's just showing a graphic or photo of the effects. 3.6 mil here, and a rather unhealthy looking 2.8. So basically what we're saying here with these cultural practices is there's 4 practices here which are fairly well straightforward and carried out very regularly. And if you tweak them a little bit, if you keep your dew levels reduced, look at your levels of rolling, lightweight rolling, nice regular light top dressing, be judicious with your irrigation practices and height of cut, your surfaces will be performing very well. They'll be firm and fast still, but with all these various tweaks to these areas here, your levels of disease will be significantly reduced. Okay, nutrition, as I said, is crucial for healthy plants. Apart from supplying nutritional needs of the plant, there's 8 of the essential elements actually have a direct effect on disease in turf grasses. We're not going through them all today, but as an example, we look at nitrogen. Like I said before, spoon feeding of nitrogen during periods of high anthracnose levels will help to reduce this disease. And this graphic here from Rutgers again, now I'm not suggesting these are the levels of nitrogen you should be applying, but it just highlights the effect nitrogen actually has on anthracnose levels. This here is the low level of nitrogen. You can see the high level of disease, and increased nitrogen reduces it. So spoon feeding of N products through these high levels of disease pressure will help to reduce anthracnose. Again, this is another very commonly used compound, ferrous sulfate or iron. For generations we've been told to use it. It'll reduce disease, microdocium especially. But really up to now there's been no solid scientific data to support this. So this is again from ICL at the STRI trial areas. And interesting, as you can see, this light blue area is the level of disease on the untreated plots. Applying this chelated iron product preventatively has some effect on disease levels, okay, but interestingly enough, applying it at first signs of disease as a curative effect had a very good effect on reducing microdocium, significantly lower than the others, the untreated control or the preventative control either. So applying ferrous sulfate or liquid chelated iron at signs of first signs of disease will help to reduce microdocium as well. The next compound we're going to look at, or element, is silica. Now, we've been using silica in the greenkeeping business really for the last number of years. It's applied anecdotally to increase the turgidity and speed of the greens. But with regard to disease suppression, what will happen is if you're regularly applying silica, it will build up in the outer layers of the cell walls. Now, anthracnose has to penetrate the leaf of the turfgrass by forming what's called aprosoria, and these actually burst through the outer layer. Both anthracnose then and microdocium, when they're in the plant, microdocium actually infects via the stomata, but both of these pathogens, once they're in the plant, will enter the cells to extract nutrients. So again, silica can help to reinforce or strengthen these cell walls. Silica also has a secondary effect, as a direct effect on fungal growth as well. And this is a Fusarium oxysporum, which is another Ascomycete fungus, which is grown on petri dishes. Now, when the media here has been moderated with silica, you can see it has a direct effect on the growth of this fungus. So apart from strengthening cell walls, silica can also have a direct effect on the fungus by slowing the growth. And this is from Lawrence Datnoff et al. They looked at the effects silica, or silicon treatments as they call them, on again on this grey leaf spot, which is a fungal disease in Florida. So the treatment levels were high enough here on the untreated, on the control plots. Silica or silicon reduced it. Chlorothalonil, the fungicide, also reduced the disease levels. And again, interestingly enough, is this combination of different compounds that had a very good effect. This synergistic effect is often seen in trials. So calcium, another very common compound, obviously is found in a lot of in high levels throughout most root zones, in fact, but it can become locked up. And as you can see here on the right, this graphic here, this is a graphic of defense responses in plants, but you can see calcium is tied up in a lot of these responses, and it's been found that elevating the amount of calcium available in the plant, increasing the elevation of concentration in the plant cells, it can cause a very significant effect during plant defense responses. So the plant actually needs available calcium in order to initiate these defenses or enhance these defenses. So making calcium available will also have an effect on your disease responses. Now this is sulfur, and this is from Clinton Mattox, who's doing his PhD in Oregon State University in the USA. Oregon actually has a very similar climatic conditions as ourselves here in Ireland and the UK. So microdocium is a very prevalent disease. Now Clint and Oregon State have been doing a lot of work over the last number of years, looking at alternative ways to reduce disease levels without using fungicides. And the first thing you'll note is this on the right. He's been looking at rolling as well, the effects of rolling on disease, on microdocium. And you can see here, without any other treatments, this here, rolling reduced disease levels compared to non-rolling. So rolling 5 days a week. So he looked at rolling and non-rolling on his treatments applications also. So over here on the left, you can see sulfur had a very good effect on the reducing microdocium. This PK Plus is a phosphite product. Again, a very good effect at reducing microdocium. And again, you'll note this combination of different compounds, the sulfur and PK Plus phosphite, very effective. It almost eliminated totally the microdocium in this particular case. Regarding sulfur, they recommend a 98 kgs per hectare per year applied monthly through September up to March. But they found sulfur with a phosphite product very effective actually at reducing microdocium. So this is some of the more latest research from Oregon State. And thanks to Gold Crop in Ireland for this information here in these slides. This in fact was a Simplot protocols which they're testing over in Oregon State on their trial areas. As you can see, they have a big area to work with. And basically what they're looking at is combinations of various compounds to see how they can reduce microdocium. Now the first plot picture you should look at is this number 1 here. This is the untreated control, and you can see there's a very high level of microdocium there, very nice indeed. And interestingly here at number 7 is the fungicide with tebuconazole. Not that effective really at reducing it, but all these other photos here of plots are basically combinations, different combinations of sulfur, of phosphite, extreme green, which is a sulfur iron product, and a pigment. So they're looking at different combinations of rates and different compounds together, but basically they're all sulfur, phosphite and iron, and as you can see they're very effective at reducing disease. This is another replication of those trials. Again, you can see number 1 is the untreated control, and the fungicide again not very effective at reducing disease. But again, as you look through all of these here, sulfur, phosphite, iron, sulfur and pigment, all very effective at reducing disease. As you can see here, this is a treated plot here with it has sulfur, durophyte and extreme green, and you can see the untreated plot next to it here. So you know the name of this presentation is suppression of disease. Can we do it without fungicides? And it looks as if that we're getting there, all right, there's no doubt about it. And this is just a good contrast between the untreated microdocium affected plot and this plot with phosphite, iron and pigment. So this is very exciting, I think, and really good results from Simplot and Gold Crop over at Oregon State. So back to our own trials. Like we saw the effect of our treatments regarding microdocium and anthracnose. So okay, we're getting a suppression in the field, but then we had to figure out how was this suppression coming about. So I had a large number of greenhouse pots and samples to work with, and we used a whole range of laboratory studies to figure out what was going on actually. As you can see, we had plenty of various pots and poor turf grasses which didn't survive a lot of the time. There were all sorts of things happening to them. The processes we used were quite interesting. One we used was fluorescent microscopy. This looked at the infection processes and the defense responses in plants. As you can see on the top left, this would be a soil sample taken from an infected area. What we use actually is a stain which attaches to the fungal cell walls, and when viewed under a UV ultraviolet light microscope, it fluoresces, and you can clearly see the hyphae or the mycelium of the fungus. So using this procedure, we're able to track the infection process right through from the soil as it grew up over the base of the plants. This here is a leaf, an infected leaf covered with a dense mycelium. We then tracked how it got into the plant and how it went through the plant and how the plant then responded. And at the end of infection, the hyphae exited, and you can see here on the very right hand side, this is the mass of reproductive structures, the conidia, which are being ejected from around the infected leaf. So we looked at the process of infection, and then the process of plant defenses. We used different stains and different procedures to see how they responded. This here, B for example, is an infection site where the microdocium would have entered, and this burst of blue is actually part of the defense process. This is actually hydrogen peroxide as part of what's called the hypersensitive response. Again, here on this photo here, you can see the stoma being infected by the mycelium, And again, this area here of hydrogen peroxide as a defense response. So using these, we tracked the infection process. I mentioned this one first. We looked at the direct effect phosphite actually had on the mycelial growth of microdocium. So we had our microdocium samples like this here, which we grew on petri dishes, and these had been amended with various compounds and various strengths and rates of compounds, particularly phosphate. And we found, in fact, that if you look at this would have been day one here on the left. These plates would have been inoculated with microdocium, and after 4 days, this is a phosphate treated plate, and this is a control plate. So after 4 days, this piece of mycelium would have grown out to the edges almost, whereas in the phosphite treated PDA, the growth was slowed down significantly so. So the phosphite was having a direct effect on the growth of the mycelium. However, the main area of suppression, in our opinion, was in fact the infected plants respond with complex chemical defenses, and it was these defenses that were being manipulated by the phosphite treatments. Just to give a brief outline of plant defenses, over here on the right, this is the plant here. Now the pathogen would enter the plant into the cell, and as soon as the plant would recognize that it's been infected, it would have some short-term defenses, such as what's called reactive oxygen species are generated, and of these, hydrogen peroxide is a key component. So this would be quite corrosive and would attack or would affect the actual pathogen, but also it would act as a signaling compound, so that the cells here surrounding the infection site would actually be programmed to die. The idea being that while the pathogen was here extracting nutrients, these cells here would be dead, and it would sort of starve out the pathogen basically. So while this first short-term defense was being carried out, other compounds were being produced and traveling through the plant to produce more long-term defenses. Salicylic acid is one which would be produced, and this travels through the plant and initiates defenses. The defenses would include these phenolic compounds, which cover a wide range of varying compounds. Some would actually strengthen cell walls, and some would produce what's called phytoalexins, which are like us producing antibodies. So when we're infected by the flu or a virus or whatever, our bodies will respond with antibodies to try to resist this infection, and plants are much the same. They would send out these signaling compounds, and other parts of the plant would then respond with defense compounds. And of course the big question was, can these defenses be stimulated or enhanced? So can we sort of inoculate our plants prior to infection, and can we help this response during infection so that they're better able to respond to defense to infection? And there's 3 compounds here which so far are proven successful in this area. Civitas is one, which is not available in the UK or Ireland, but it's a Petro-Canada product which is very successful in the States and in Canada. And basically it works by inducing these defenses prior to infection. So you apply Civitas to your plants, your turf grass, and this induces these defenses, and when the infection occurs, these plants are much more able to respond. So it's almost like getting the flu jab and it working correctly. Now salicylic acid, as I said, is a signaling compound which induces defenses throughout plants, and it's been shown that applications of this can in fact induce defenses and reduce disease. Again, we're going to look at phosphate now, some work I did on it, and we'll look at that now in a few moments. So here's this what I was saying about salicylic acid. This graphic here shows exactly what's going on. So this leaf on the left would have been infected, for example. So while this short-term defenses were going on, salicylic acid is translocating through other parts of the plant which are not infected, and inducing these compounds for defense. And it has been found that applying salicylic acid will induce these defense prior to infection and lead to less less infection being evident. And some compounds or some products these days now are starting to include salicylic acid as part of this defense induction. So anyway, like I said, phenols or polyphenols are crucial for defense. There are a wide range of compounds. Part of our research, we looked at infected and non-infected plants over 3 years, turf grasses. This is Poa annua and creeping bentgrass. And what we found over the 3 years in all cases, well, not in all cases, but the majority of cases, was that the infected plants displayed higher levels of polyphenols each time. So this indicated to us that indeed, yes, polyphenols were crucial for plant defenses. And the next stage we looked at was in the greenhouses, again with Poa annua and Agrostis solanifera. So we applied phosphate and phosphite over 6 months sequentially by standard rates. And at the end of the 6 months, we found that there were significantly higher levels of these polyphenols or phenolic compounds in the phosphite treated plants compared to either the control or the phosphate plants. So this was showing us, in fact, that apparently applying phosphite will actually prime your plant with these defense compounds, so your plants will be more ready for infection, or in fact actually ready for abiotic stress either, as the responses are quite similar. So we then looked at how it affected infected plants in the greenhouse, and this is a sample here. This is Poa annua here. So we had pre-treated these plants in the greenhouse with phosphate and phosphite, and then we inoculated them with microdocium here on day 0, and over the 10-day period we took samples and measured the levels of phenols in these plants, and you can see there's significantly higher levels of these defense compounds in the phosphite treated plants. So regarding microdocium suppression, some of our studies found that phosphite has a direct effect on the mycelial growth and also disrupted the morphology of the fungus, which would actually lead to it releasing elicitors which would be picked up upon by the plant. But the main area of suppression was in this priming and enhancement of these defense responses. Phosphite primed the plant prior to infection and enhanced not only phenolic production but also hydrogen peroxide in the short term. So it's this enhancement of defenses that caused this suppression of microdocium in the field trials. So what about the anthracnose study which we did last summer? How did we get this amount of suppression, not only with phosphite but with this cocktail of nutrients? Well, first of all, the phosphite obviously induced the defenses in the treated plants, and we had a good reduction from that. But this one here, with this cocktail of compounds and products, were very effective. So how did this come about? Well, first of all, one of the components, this plant food 29, actually contained, apart from nitrogen, which was spoon feeding, and as we saw, spoon feeding of nitrogen will help to reduce anthracnose, but also contained calcium, sulfur and copper. Now all these are key to plant defense responses. Another product we use contained polyphosphate and also potassium phosphate. So again, that was adding to the defense responses. And then we had two other products then were actually increasing calcium availability and also providing salicylic acid. So what we've seen the last few moments regarding plant defenses, we're supplying all these components to these treated plots. So we were reducing stress first of all by providing balanced nutrition, but we're also providing phosphate, calcium, sulfur, copper and salicylic acid. So these all combine to help reduce this level of anthracnose. So just the take home points first of all is, as turf managers we need to look at this disease pyramid and see what factors we can actually influence ourselves to reduce disease pressure. We mentioned, dew removal, air movement, all these environmental factors. The cultural practices we saw have a good effect on reducing disease: rolling, sand top dressing, judicious mowing and irrigation practices. Again, we looked at the plant factors that use less susceptible species if you can. But obviously the balanced nutrients inputs are vital, ensuring your plant is in a healthy state and unstressed as possible. And again, the judicious use of various compounds will not only reduce disease incidence, but they can be used as defense activators to enhance these responses. So that's about it really. Just that none of these that we've mentioned will eliminate disease fully, certainly not on their own. But if you can put them into a program of these various nutrients and elements and cultural practices, you will reduce disease incidence. And not only that, but it will enhance your fungicide program if you have any fungicides. So that's about it really. Hopefully now, if you want to find out more, you can follow me on Twitter. I'm always on there giving out information or whatever. And, hopefully now you found this of interest. It's my first recording of a presentation, so hopefully it went okay. So thanks for tuning in, and hopefully this was informative and helpful to you.